divisions. So before going to the chromosome, we should know something about the chromatin. So you have already heard the term chromosome. You have already heard the term chromatin and the DNA. So what are the difference between this chromosome, chromatin and the DNA? So now get into this. So in the cell cycle, let's suppose this is the cell cycle. So in the cell cycle, there are the different types of the phases are present. So the first stage is called as the gap 1 phase, or it is also called as the G1 phase. So that means, and before that, another stage is called as the G0 or the quiescent stage. So that means if any cell, if it wants to go for the division, then it transfers from the G0 to the G1. If it doesn't want to divide, then it remains in the G0. So that's why the G0 is called as the quiescent stage. So now, if the cell wants to divide, then it left the G0, then it transfers to the G1 and the gap 1. Then after the gap 1, there is a phase is called as the S phase. S for synthesis. So what is the synthesis, or why the name is called as the S phase or the synthesis phase? Because here the what does the it synthesize? It synthesizes the DNA. So that's why it is called as the S phase. So after the S phase, another phase is called as the gap 2 or G2 phase, and after that, that is the main phase, that is called as the main phase or the M phase, or it is called as the mitosis or the meiosis phase, because in this phase actually the cell division occurs. So the gap 1 S phase, gap 2 is called as the interphase, and this is called as the M phase is called as the division phase. So what is the difference between the interphase and the division phase? Because in the interphase or during the interphase, the cell already prepares for the division, but in the interphase there is no division in the cell. The cell divides only in this M phase, but in the interphase it prepares for the division. So that's why it is called the interphase. Okay. So now come to the term the chromosome, chromatin and the DNA. So what is the DNA? So DNA, simply imagine a thread of the, simply you can imagine like the thread. The thread is like this. So if this thread, it coiled together one and more, and now it forms the structure like this, and this is called as the chromatin. Okay, so in the chromatin at the middle it is called as the centromere. So that means our centromere, it is also called as the primary constriction that divides these two arms. This is the one arm, this is the another arm, and this is called as the chromatin. So that means in the gap 1, the structure of chromosome, it looks like this, and this is called as the chromatin. Okay, so that means what is the difference between the DNA and the chromatin? That means the single DNA or the one DNA coiled together each other, and it forms the chromatin like structure and looks like this. That means one chromatin, we can say that one chromatin contains one DNA. So that means at this page, that is, there are 23 DNA are present. So that means the 23 chromatins are present. So in our cell, for example, in the animal cell, the 23 DNA are present, and this is called as the chromatin in the gap 1 phase. Okay. So now in the S phase, the DNA is synthesized. So that means another DNA, that means in that chromosome, one DNA is present and it called together to form this like structure that is called as the chromatin. So in the S phase, one chromosome or the one DNA replicates and now two chromosomes, so two DNA are formed. So that means one chromosome again coiled together to form the chromatin. So now this DNA also coiled together and now it forms like this. That means the two chromatins here, two chromatins are formed here. That means in the S phase. So that means in the S phase, two chromatins are present and now these two chromatins are joined and it looks like this. So that means this is the first one, this is the second chromatin. They are joined together to form in the S phase. So now this one is called as the sister chromatid to each other. That means this one is called as the chromatid. This one is also called as the chromatid, and these two chromatids are also called as the sister chromatid to each other. So that means during the S phase, the chromatin looks like this. That means in the G1 phase, the chromatin looks like this. That means in that one DNA is present, and from the one DNA, it forms one chromatin, and at the middle, it is called the centromere. In the S phase, because the DNA is synthesized, that means the DNA replication occurs. That means from one DNA, two DNA are formed. And from the two DNA, obviously two chromatins are formed, and two chromatins are again joined together. So now each one is called as the chromatids. This is the one chromatid, this is the another chromatid, and they are called as the sister chromatids. So that means the chromatin in the S phase, it looks like this. But the chromatin, the look in the G1 looks like this. And what is the difference between the chromatin present in the gap 1 and the S phase? In the gap 1, the chromatin contains one DNA. But in the S phase, the chromatin contains two DNA. So that means in the cell, for example, in the human cell, at the gap 1 phase, there is the 23 DNA are present. But in the S phase, 23 chromatins are present. 23 chromatins are present, but the DNA are now the 46 DNA. But here, the 23 chromatins are present and the 23 DNA are present. And the structure looks like this. But in the S phase, the chromatin, same number of the chromatin, that means the 23 chromatins, but the 46 DNA, that means the DNA number doubles. So, two chromatids are formed and each are called as the chromatids or the sister chromatids. So, in the gap 2 phase, again the same like structure as present here. But in the division phase, the same like structure are present. Look at this structure. This one, and this one. What is the difference between this structure and this structure and this structure? So, during the interphase, that means in the S phase and the gap 2 phase, same chromatin structure are present, but this chromatin is present in the thin structure or decondensed structure. But in the M phase or during the division phase, now it is a very, very thick. So it is also called as the thick or condensed. So this is the difference between the chromatin and the chromosome. And because this thick or the condensed structure of the chromosomes are present, so condensed chromatin are present, now you can say that this is the chromosome. So that means the chromosomes are actually the chromatins, but they are present in the thick or the condensed structure, and now it is called as the chromosome. So that means this is the chromatin. That means the one chromatin contains one DNA. At the S page and the gap 2 page, this is also called as the chromatin, but it contains two DNA, and these chromatins are present in the very thick, very thin, or it is called as the decondensed manner. But after going to the M page, it is very thick, and it becomes very thick or the condensed structure, and now this is called as the chromosome. So that means what is the difference between the chromosome and the chromatin? The chromosome is the thick and the condensed structure that is present during the M phase. Or the chromatin is the structure that is the thin and the decondensed structure that is present in the interphase. And what is the DNA? DNA forms the chromosome. One chromatin, the one DNA forms one chromatin in the gap 1 phase, and in the S phase, because two DNA are formed, so one same chromatin, it contains two DNA. One contains, or it is called as the chromatids or sister chromatids. So the two sister chromatids are found in the one chromatin, and here also same structure. That means here also one, you can say that now it is the one chromosome, but it contains two DNA. This is this one, and this is the another one. This is the one chromatid, this is the another chromatid. These two chromatids are called as the sister chromatid. So this is the difference between the chromosome and the chromatin and the DNA. So this is the all about the difference between the chromosome, chromatin and the DNA. So now come to what are the different types of the chromosomes. So whenever I say the chromosome, so you can imagine that that is the chromatin that is present during the M phase or the division phase. So now come to the types of the chromosome. Types of chromosome. So there are the four types of the chromosomes are present. One is called as the metaphase. Second one is called as the sub-metaphase. Third one is called as the acrocentric, or it is also called as the metacentric. You can say that simply it is called as the, okay, you can, okay, different types of chromosome. So first one is called as the metacentric. The second one is called as the sub-metacentric. Third one is called as the acrocentric, and the fourth one is called as the telocentric. So these four types of the chromosomes are present on the basis of the presence of the centromere. So look at this structure. They are the different types of the chromosomes. Look at this metacentric chromosomes. So in the metacentric chromosome, this is the centromere, or the centromere is also called as the primary constriction. Primary constriction means here it forms a gap, or it divides these two arms of the chromosome. So this is the centromere, and this metaphase or the metacentric chromosome, the centromere is present at the middle. So that means at the metacentric, the centromere is present at the middle of the chromosomes. So that means this is the one arm, this is the another arm of the chromosome. So now because the centromere is present at the middle of the chromosome, these two arms are present they are of the equal size. Okay, so this is the metacentric chromosome. So next come to the submetacentric chromosome. This is the submetacentric chromosome. So here the centromere displays at the away just after the middle. That means actually the middle part is the here. But the centromere is present away, some part away of the middle part. So it is the away from middle. So because the centromere is present away from the middle, so now these also two arms are formed, but these two arms are unequal size. Unequal size. So that means one arm is the shorter. So here this one is short and this arm is a very long arm is formed. So always this short arm is called as the P arm and the long one is called as the Q arm. Okay, so because now the centromere is present away from the middle, two types of the arms are present. One is called as the short arm, another is called as the long arm. The short arm is called as the P arm and the long arm is called as the Q arm. So this is the submetacentric chromosomes. So again another is the acrocentric. This is the acrocentric. So acrocentric means now the centromere displaces near the terminal part. This is the terminal part. So that means a very large arm or the long arm or a Q arm is formed and a very short arm or the P arm is formed due to the acrocentric chromosomes. So another is the telocentric. The telocentric means the centromere transfers exactly to the terminal part. So terminal part. So that means after the transferring the centromere to the terminal part, so now only one arm can be formed and this arm is called as a Q arm. Why? Because already it is very long. So this is the different types of the chromosome on the basis of the presence of the centromere. So that means in the metacentric, the centromere is present at the middle. So after that, or because of that, the equal size of the two arms are formed. But in the submetacentric, acrocentric and the telocentric, unequal size of the arm is present. The short arm is called as the P arm, the long arm is called as the Q arm. So in the submetacentric, the centromere is present just away from the middle. So because a P arm and the Q arm is formed, the acrocentric, the centromere is near the terminal, not exactly to the terminal, near the terminal. That means a very long arm is formed and a very short arm is formed. So at the telocentric, the centromere is present at the terminal. That means one arm is formed and that arm is the long arm, or you can say that it is the Q arm is formed. So these are the different types of the chromosome on the basis of the presence of the centromere in the chromosome. So another chromosome is present that is called as the satellite chromosome or the satellite chromosome, or you can say that SAT chromosome, which is the same, that is the satellite chromosome. So what is the satellite chromosome? One is the chromosome and is the satellite chromosome. So what is the difference between that? Look at the structure of the chromosome. This is the chromosome, simply chromosome, and look at the structure. And this structure is called as the satellite or the SAT chromosome. So what is the difference between the structure of this chromosome? So in the satellite chromosome, one centromere is present or the primary constitution, which is present also in the chromosome. So the main difference is this structure. So that means this is the one constriction is present in the obviously in the chromosome here also, and that is called the primary constriction. But some chromosome contains another constriction here. So because of that, of the presence of this constriction, one this like structure is present, and this structure is called as the satellite. So in the satellite chromosome, a secondary constriction is present, and this because of this secondary constriction, it forms a satellite, or you can say that a small fragment are formed. Here also some again another fragment is formed, which is looks like the satellite. So that's why in the satellite chromosomes, because of the presence of the secondary constriction, the satellite of the small fragment is formed. So it contains secondary, or the two constrictions are present in the satellite chromosomes. Not all the chromosomes are the satellite chromosomes. Some chromosomes contain satellite. Which chromosomes? So in human being, it is the 13, 14, 15, 21 and the 22 chromosome. So these 5 chromosome forms the satellite or the secondary constriction. So that means these exact 5 chromosomes are called as the SAT chromosomes or the satellite chromosomes. देखिए एक टॉपिक है कार्योटाइप। कार्योटाइप तो आपके मन में आया होगा कि कार्योटाइप एग्जैक्टली क्या है। देखिए कार्योटाइप को हम बोल सकते हैं कंप्लीट सेट ऑफ क्रोमोसोम, कंप्लीट सेट ऑफ क्रोमोसोम इन एन ऑर्गेनिज्म। तो मतलब एक ऑर्गेनिज्म में जितना क्रोमोसोम है उसका कंप्लीट क्रोमोसोम स्ट्रक्चर को अगर देखेंगे इसको हम बोलेंगे कैरियोटाइप। देखिए इस स्ट्रक्चर को आप देखिए, इस टोटल स्ट्रक्चर को हम क्या बोल सकते हैं, कैरियोटाइप। ये किसका है, ये ह्यूमन का है। और ह्यूमन में कितना क्रोमोसोम है, 46 क्रोमोसोम, और दैट टू 23 पेयर में है। तो पेयर वाइज वो और 46 क्रोमोसोम मतलब 23 पेयर, मतलब इस 23 क्रोमोसोम जो है, इस 23 क्रोमोसोम को आप इसे देख सकते हैं 1 2 3 4 5 6 7 8 9 10 11 12 13 14 15 16 17 18 19 20 21 22 और एक 23, ये हो गया 22। 23 मतलब एक हो गया मेल का, एक हो गया फीमेल का। अगर फीमेल होगा तो ये 22 तो होगा, उसके साथ ये वाला होगा, ये हो गया 23 हो गया। अगर मेल होगा तो ये होगा, उसके साथ ये वाला होगा, तो ये हो गया कैरियोटाइप और द कंप्लीट सेट ऑफ द क्रोमोसोम इन ऑर्गेनिज्म। तो जिसका जैसा हो गया, मतलब ह्यूमन का 46 क्रोमोसोम है, मतलब 23 पेयर है। जो 23 पेयर को एग्जैक्टली कैसे सजा जाता है, इस सजावट को ही बोलते हैं कैरियोटाइप। आप पहले जान लीजिए वो कैसे सजावट होता है, मतलब ये जो हो गया वो कैसे हो गया। देखिए एक सेल डिवीजन में एक फेज आएगा, वो फेज को हम क्या बोलेंगे, मेटाफेज। मेटाफेज में क्या होगा, मेटाफेजिक प्लेट एक बीच में होगा। इस बीच मेटाफेजिक प्लेट में वो क्या होगा, ऐसे ही क्रोमोसोम बीच में रहेंगे। उसके बाद यहाँ पर क्या होगा, स्पिंडल फाइबर फॉर्मेशन होगा। तो इसको बोलेंगे क्या, मेटाफेज। तो मतलब मिडिल पर, मतलब सेल के मिडिल एकदम मेटाफेजिक प्लेट पर ऑल क्रोमोसोम, यहाँ पर एक मतलब एक ही मेटाफेजिक प्लेट में सभी क्रोमोसोम रहता है। तो इस स्टेज को हम बोलेंगे मेटाफेज। तो इसी स्टेज को हम क्या करेंगे, तो अभी उसको स्टेन कर देंगे। स्टेन मतलब क्या होगा, अगर स्टेन कर देंगे किसी डाई पर, अगर स्टेन कर लेंगे वो क्या होगा, एग्जैक्टली जहां पर होगा उसका स्ट्रक्चर वो आ जाएगा, मतलब वहां पर रुक जाएगा, स्टेन हो जाएगा। किस स्टेन कहां पर होता है, कैसे होता है, डाई पर। और एग्जैक्टली बोलेंगे किस डाई पर होगा, जिम्सा, GIEMS है, जिम्सा एक डाई है, उससे वो स्टेन होता है। वो कहां पर होता है? मेटाफेजिक, और मेटाफेज स्टेज पर ही वो स्टेन हो गया। तो अभी उसका हम आ गया, ऐसा स्ट्रक्चर आ गया। अभी क्या करेंगे? उसको फोटोग्राफ मतलब सिंपली फोटो निकालेंगे। तो जब फोटो निकालेंगे इस स्टेज पर, इस मतलब इस फोटो उठाने का काम को हम क्या बोलेंगे? इसको हम बोलेंगे पिक्टो माइक्रोग्राफ। पिक्टो माइक्रोग्राफ, पिक्टो माइक्रोग्राफ क्या हो गया? पिक्टो माइक्रोग्राफ क्या हो गया? जो मेटाफेजिक प्लेट में, मतलब मेटाफेजिक स्टेज पे जो स्टेन कर दिए, उसके बाद जो फोटो उठाए, इस फोटो करने का प्रोसेस को ही हम बोलेंगे पिक्टो